Greetings, my name is Benalcazar, and I am going to present the research paper, a 3D iris scanner from a single image using convolutional neural networks. This work has been published in the IEEE Access Journal as a joint research between Universidad de Chile and the University of Notre Dame. The objective of this research is to capture a 3D representation of the iris surface from a single visible light image. The outline of this presentation is the following. First, I will discuss the motivation behind the development of this method, as well as possible applications. After that, I will present previous works and how we propose to improve upon them. Then, I will introduce the proposed methodology and discuss the results to finalize with the conclusions. The motivation. There is a high demand for ever more accurate methods to recognize the identity of an individual. Iris recognition in particular is effective because it exploits the distinctive pattern of the human iris, which has a high variability between individuals. That is why it is used from small to large-scale applications. However, the problem with current iris recognition systems is that they use 2D images for the analysis, when the iris is in fact a three-dimensional organic structure. The human iris has two muscular systems and a sphincter which allow pupil dilation to control the amount of light that hits the retina. Those muscular systems create a relief in the surface of the iris, similar to a landscape with peaks and valleys. This is exactly what we aim to capture with our method in an iris 3D model. The additional depth information generates an extra degree of freedom, which is expected to increase iris recognition performance even in larger databases than currently possible. Additionally, an iris 3D model has potential applications in the detection of closure angle glaucoma, a disease currently diagnosed with an optic coherence tomography (OCT). A healthy iris is flat, and it produces a good circulation of aqueous humor. However, in this disease, there is a blockage, intraocular pressure builds up, this curves the iris and also damages the cells in the retina. Due to that, the patient can start to lose their sight. Ophthalmologists use an optic coherence tomography to detect the presence or absence of this curvature. However, this is a very expensive equipment and exam. The proposed method can deliver similar information using simply a cell phone camera. The related methods. There are few publications in the area of 3D iris scanning. All of them use a structure from motion to form the 3D model. This method starts by taking images from several perspectives and obtaining key points and descriptors. Then triangulation is used to form the 3D point cloud model of the iris. Finally, a dense 3D model and a mesh model can be obtained. There are few problems with this approach. The first problem is that the mechanisms to move the camera around the eye add extra complexity to the system. The need for multiple images increases acquisition time, processing time, and storage. Also, the pupil could dilate from one image to the next, which can distort the 3D model. Finally, since this method depends on key points, it tends to produce more 3D points in areas of the iris with more texture, and there are even empty patches with no 3D points. On the other hand, a structure from motion is not the only alternative to obtain a 3D model from 2D images. In recent years, convolutional neural networks that can predict the depth of every pixel in an image have achieved very accurate results. They use the encoder-decoder architecture with convolutional layers. Those architectures are trained with thousands of indoor and outdoor images as the input and aligned depth maps as the target. Ground truth depth maps are obtained using laser sensors or RGBD cameras like Kinect. There are many advantages of using convolutional neural networks over structure from motion in 3D iris scanning. CNNs can produce a 3D model using just one image, therefore acquisition time and storage are decreased. Also, complicated structures for a moving camera are not needed. Then, the 3D models always get an even distribution of 3D points, because a depth prediction is made for every pixel in the input image. Finally, the distribution of 3D points does not depend on texture. Therefore, irises with high or low texture will have the same number of 3D points with no empty patches. Now, adapting CNNs for iris depth estimation is not an easy task. There are two main problems with it. First, the networks require image depth map pairs to train, and there is no such dataset available for iris images. Also, using laser sensors to obtain the depth maps is not a problem for large scenes. However, the human iris is very small and delicate. We wouldn't want to blind anybody. The second problem is that those architectures use visual cues of perspective to understand the three-dimensionality of a scene. For example, if we know the normal height of a person, when the person is big in the image, it means that they are close by, but when they are small, it is because they are farther away. With iris images, the networks cannot learn 3D information from perspective, since the iris is normalized and the only object in the scene. Those two are the main problems that we aim to solve in this paper, while keeping all the advantages that CNNs have over structure from motion. The methodology. In order to solve the first issue, that is, the lack of a dataset of iris images with depth information, we propose to use T-SquareNet, an architecture that is conceived to train with real and synthetic images. The synthetic images can have depth ground truth associated with them, and the real images help to train a GAN network that is in charge of translating the synthetic images so that they look more photorealistic. In this way, the encoder-decoder can learn how to predict the depth information of a photorealistic iris, and after training with thousands of images, the encoder-decoder will also be able to predict the depth information of real irises. In order to solve the second issue, that is the lack of perspective information in iris images, we propose to predict depth from shadows. For example, we humans can detect that this picture of the moon is not a flat terrain, but rather there are craters, and we can tell that because of the shadows they cast. In this way, we proposed to illuminate the iris not only with frontal illumination but also with lateral illumination, so that iris features and the muscular systems in the iris can cast shadows, and the convolutional neural network will be able to relate the shadows in RGB images with depth information. In order to obtain a data set with set characteristics, we propose the following device that illuminates the iris with lateral and frontal visible light. The left eye of the subject can look at printed text, and both eyes are illuminated with white LEDs with this printed circuit board. This is the lateral illumination, which is also a white LED. The acquisition device is a cell phone camera with a macro lens attached. This device can then capture close-range images of the right eye of the subject. We also modified this device so that we can take images from both eyes. Using this device, we collected a real iris dataset of 120 subjects and 26,000 images under several dilation levels. In order to obtain the virtual iris dataset, we first collected 36 OCT images from the internet so that we can have depth ground truth of real irises. After that, we used the software Blender to sculpt 100 virtual irises. For this purpose, we first placed the OCT in the YZ plane and traced the contour of the iris. After that, we created a revolution surface from this contour, and we sculpted the texture of the iris so that it resembles that of the real iris dataset. We created some virtual irises with more texture than others and with different dilation levels in order to represent what we have in the real iris dataset. After that, we produced 3D data augmentation of 4 colors, 9 positions, 5 rotations, 2 scales, and mirroring, so that we can render 72,000 synthetic images. We finally added eyelids, eyelashes, and reflections in order for the networks to learn how to segment those features. Once we acquired both datasets, we split them in 80% for training, 10% for validation, and 10% for testing. After that, we trained several state-of-the-art networks using the vanilla and full approaches described in T-squared-net. For instance, we trained the cycle gangs, dense depth, Dorn, peaks-to-peaks, and T-squared-net. We also introduced iris depth, another architecture which combines the gang of T-squared-net and the depth prediction of dense depth in order to increase performance. The results. This image represents the depth prediction test using synthetic images. Here we have the synthetic input image and the depth ground truth. From the synthetic image, gang networks such as cycle gangs, T-squared-net, and iris depth made a translation so that the synthetic image would look more photorealistic. After that, encoder-decoder networks such as dense depth, Dorn, peaks-to-peaks, and iris depth made a depth prediction from their corresponding enhanced image. Finally, we can score the similarity between the depth ground truth and the predicted depth map in order to find the performance of each network. This table summarizes the results of comparing 7,200 synthetic images for all the trained networks. The proposed network iris depth obtains the best performance in almost all the tests. For the next results, we present the 3D models obtained from human irises. The 3D models capture all the depth variations produced by iris features. In order to convey this 3D information, we have plotted a red-green mesh on top of the iris. Our 3D models have always a constant distribution of 3D points, regardless of iris texture. At a resolution of 256 by 256, the 3D models have 65,000 3D points. This number is 6 times larger than the amount of points reported in previous structure from motion-based methods. Since we segmented eyelids, eyelashes, and the pupil, those features can easily be eliminated from the 3D model in iris recognition tests. For the next test, we obtained 8 OCT slices of a subject as ground truth, 4 per eye, and we compared them with corresponding slices of the 3D models produced by the trained networks. The depth profile in the 3D model matches closely the iris contour in the OCT. This indicates that the trained network were successfully able to predict the depth of the human iris based on a single visible light image. This table summarizes the result of the comparisons. The proposed network iris depth produced a small amount of error in both the left and the right eye, and the average error is 77 micrometers. This number is 60% smaller than the amount of error produced by structure from motion, which was 123 micrometers. Finally, we propose a 3D rubber sheet model as a proof of concept. Just like its 2D counterpart, it unwraps the annular region of the iris into a rectangle that is easier to compare. However, the 3D rubber sheet has additional depth information available for the analysis. The 3D rubber sheet also helps to illustrate the amount of texture and the amount of information that can be extracted from a human iris using the proposed methodology. That is why we expect the 3D models to increase iris recognition performance. In this work, we made preliminary iris recognition tests using the 12 subjects in the test set. We have 24 eyes and 480 images for the analysis. Using this dataset, we tested using the standard 2D iris code as well as the proposed 3D rubber sheet. The 2D iris code obtained a separability index D' of 8.5. However, the 3D rubber sheet had a 48% increase in this value at 12.6. This means that we can better separate genuine from impostor comparisons. The conclusions. We developed an iris imaging device that captures images with lateral and frontal visible light. We acquire a dataset of 26,000 real iris images and 72,000 synthetic iris images. With those datasets, we trained several state-of-the-art networks and developed IRIS-Depth, a network that combines T-squareNet and dense-depth to increase performance. The 3D models in our method have 65,000 evenly distributed 3D points, regardless of iris texture. IRIS-Depth obtained 77 micrometers of reconstruction error, which was the smallest between all the networks and structure from motion. Finally, iris recognition performance increased in 48% using the proposed 3D rubber sheet. This illustrates the potential applications of 3D iris scanning in biometrics. This concludes my presentation. Thank you for your attention. These are the references relevant to this presentation.